Okay, so hi everyone. My name is Tim. I am a lab tech here in the academic upgrading department, and we're doing a lab today. Unfortunately, I don't fit into the frame fully. Today is looking at indicators and how we can use them to help us distinguish acids and bases from neutral solutions. That is, those solutions that are neither acidic nor basic. And what is the goal or point or task that we have in this lab? Well, it's basically to determine, using some indicators, whether a variety of solutions, some of which may be familiar to you, are acidic, basic, or neutral. That is, we're not just guessing here, although we're going to make a prediction. You already probably have made it. If not, you can do it later. And we're going to test that prediction basically and see whether our prediction or your prediction was correct or not. So we're going to draw some kind of a conclusion as well. Sodium hydroxide aqueous dilutes HCl aqueous. So those two, we know that one of them is a base and one of them is an acid. So that is something that we know for sure. And these two guys, essentially, we're going to use them as positive control. That is something that we know positively, with 100% surety, the chemical nature of. And we'll see how different indicators respond to the known acid and the known base first. And then we're going to use that knowledge and observation to determine, using those same indicators, whether grapefruit juice and everything that's listed under it or below it is an acid or the base. So that's kind of the big plan for today. Substances. The first one is distilled water, sodium hydroxide, and then HCl. So this is what I'm going to dispense here. I'm going to grab some distilled water, and I'm going to dispense some water into the first row of the wells there. So I'm going to add some water here. Okay. The second row of the wells, we're going to add sodium hydroxide. Now, sodium hydroxide, you see that we know that it's a base, right? Because it has a wording sign, and the chemical formula probably tells you the same. If I just spoiled your prediction, sorry about that, but I'm sure that by now everyone probably knows what sodium hydroxide is. And like I said, the whole point of including sodium hydroxide and hydrochloric acid, that also gives it away, is to use them as positive controls. That's something that we know the chemical nature of without any doubt. So this is hydrochloric acid, and that acid part also gives away what it is. I'm going to add it to the last row on this 3x4 plate. So right now we have the same arrangement as you have in your table. Distilled water first, sodium hydroxide next, HCl. And I'm going to add the indicators, or start adding the indicators. And I'm going to show the indicators to you very briefly what they are. So we have blue litmus. Now, you see that this is kind of it's called blue litmus, but it's slightly purplish in its sort of native state. Phenolphthalein is colorless when dissolved in water alcohol mixture. Nothing remarkable about it until we mix it with something. And bromothymol blue, you see that it's a very nice clear blue. So this is a difference for you, but this is synthetic indicator, and this is derived from a plant. So you see the difference, even though they both have blue in the name, the color difference of the synthetic color versus the natural color is quite different. But it shouldn't really matter because we know what the color change of each indicator is. Or if you don't know what it is, we'll see when we test these first three substances. So let's add the indicators to distilled water first. Oh, and I didn't show you the last one. The last one is pH paper. And pH paper is actually just some plain paper that's been soaked up and dried in a solution like that one, but of a special indicator that not only changes color, but you can also use the color change to predict or tell what the pH of the solution is. And the way this works, you dip the strip into your solution, or you apply a drop of the solution on the strip, and then you quickly compare the resulting color and read off the pH value of that solution off this scale that's printed on the vial or this jar. And that's how it works. So it's really convenient because you can tell right away what the pH in pH units is of your solution. So this blue litmus. Then I'm going to add phenolphthalein to the next one. And I'm going to add bromothymol blue. What I need here is something to stir it with, because this by itself does not provide for a very nice stirring. So hopefully you can see. For your observation, just record the color. Don't try to make any conclusion just yet. So you see that here you have, so this is our water, right? And we have blue litmus, and the color is, you can write it down as blue, or purplish blue, whatever. Phenolphthalein colorless, and bromothymol blue is blue. Now I'm going to apply a little bit of water to this strip of indicator paper. Let me just grab my tweezer. I'm going to dip this strip of paper into the water, and then we're going to look closely at the resulting color and see what the best match is. Oops. There color-wise, and I think that's due to the camera. So if I look closely, it actually looks more like between 7 and 8, right? Because you see that 12 is printed over the deep blue, right? So it's definitely not that. So it may be 6, but it has some greenish hint of green to it. So I would say this is probably between 7 and 8, more close to 7. I would write down it as 7. Let's do the same thing, but now with sodium hydroxide. And watch as the color change as I add these indicators. Let me see. So this is all distilled water. You can ignore the slight yellowish color here because that's where I dipped the indicator strip, and I do sodium hydroxide next. And then I'm probably going to add everything without commenting, and then we're going to look at all indicators at the same time, just to make sure it doesn't take too long, because I don't know how much time Dan has planned for this. So, okay, that was quite a dramatic change there. Now let's look at this again. So that second row of wells, this is what we're looking at. So this one. So we have blue again for bromothymol blue. It's you can ignore the slight differences here. This appears as more purple in distilled water, and this appears as quite blue. You can still write blue because we don't really use gradations for this indicator of color. And phenolphthalein with sodium hydroxide is deep pink, almost red to me. and bromothymol blue remained blue. And let's see what the indicator paper tells us. So I'm going to solution, and bam, that changed to a really deep color. So I would say this is something, you know, it's kind of displaying everything to me in a mirrored pattern, maybe 11, 12. Yeah, probably 11. Sodium is hydrochloric acid. Hydrochloric acid is also a colorless liquid by itself, nothing remarkable about it. But when we mix this acid with our indicators, we'll see what happens. I've already added acid there, but let's add the indicator. So I'm going to add the litmus first. Then I'm going to add phenolphthalein. And then the bromothymol blue. Again, if we look at these samples here, the last one that's closest to me, you see that this bromo blue litmus turned red. It may look orangish to you, but it's actually reddish. Phenolphthalein remained colorless, and bromothymol blue turned yellow. Let's use another strip of the universal indicator and see what it tells us. Again, quite a dramatic color change, which to me looks like somewhere between 1 and 2. You can write probably closer to 1, but you can write either. So this is hydrochloric acid universal indicator. So we have tested our first 3 substances, and one of them was water, which is pH 7. You saw how the indicators responded. Yeah, probably 1. And then we tested a known base, sodium hydroxide, and a known acid, hydrochloric acid. So, and we saw the color change. Now we can use this knowledge how each indicator responds to neutral, acidic, and basic solutions, and try to classify some of the household type substances as either acidic, basic, or neutral. So the number 4 grapefruit juice, this is going to be glass cleaner, and then this number 6 soap solution. So I'm going to put it this way here, because unfortunately I'm kind of add some. It has a slight hint of pink to itself, but it's probably fine. It shouldn't affect our results too much. If anything, we can add some water and dilute it. Okay, so that was grapefruit juice. Then I'm going to add glass cleaner to the next set of wells there. So glass cleaner, now you may or may not be familiar with this. You see that also is has a slight bluish color to it, probably so people don't ingest it by accident. But I personally don't know anyone who has ever attempted, but who knows. So this is the glass cleaner, and sometimes it's also known under the name ammonia cleaner. So that gives you any idea about its chemical nature as far as the prediction goes. And you can use that knowledge. And soap solution, again, just your typical hand soap, not the maybe aggressive shop type industrial soap, just one of those greenish soaps that you may have sitting on your sink. I added some water so it's not as thick, right? So it mixes easily with the indicators, but it shouldn't change the chemical properties significantly. Blue litmus into the first well of the grapefruit juice. Then goes phenolphthalein. Finally, bromothymol blue. And lastly, let me just add a bit more litmus. I think I used too small of a drop. And lastly, universal paper. But before I dip that universal paper into the well, let's look at the color change. So this is grapefruit juice number 4. The first one is blue litmus and it turned red. Phenolphthalein remained colorless. There's a hint of pink, but that's the color of the grapefruit juice itself. Bromothymol blue turned clearly yellow. And let's dip our indicator paper into this grapefruit juice, and I'm going to have this. Now, we kind of need to work quick. That's why I don't pre-mix it sort of with our test substance. So let's look at the, let me just distribute this evenly. Let's look at the pH chart. What does it look like to you? Probably 3. Yeah, maybe like 3. So you see that we had what? 1 for hydrochloric acid, and we have 3, pH 3, for our grapefruit juice. So pH 3 is quite a bit away from 7, which is neutral. And now you probably know why you can't really drink grapefruit juice sometimes undiluted, because it's what? It's so acidic, right? Depending on the cultivar. Exactly. So let's look at number 5, which is glass cleaner. Again, this can be a bit of a mystery to some people because if you're not familiar with what exactly it is, but I mentioned ammonia, so that might give it away as far as your prediction goes. But let's see what happens when we add the indicators. Again, starting with the blue litmus. Then phenolphthalein. Finally, bromothymol blue. So let's look at what we have here color-wise. So you see that where I added the indicators, we have blue here with the bromothymol blue. We have pink color developing with phenolphthalein, and we also have blue with bromothymol blue. So both blue litmus and bromothymol blue gives us blue color when mixed with ammonia or glass cleaner. And we have a lighter pink with phenolphthalein compared to what we had with sodium hydroxide, if you remember. You see that with sodium hydroxide, that pink was so deep that it was almost blood red. So what does it tell us about the chemical nature? Well, you be the judge and fill out your conclusion eventually accordingly. But dip one of these strips into the glass cleaner. Let's see. Again, dramatic color change. Let me just sort of let it soak up the liquid. Let's look. It's not as that deep. It's probably not 11, but I would say this is between what? Maybe 9 and 10. Yeah, so maybe 9 and 10. Probably closer to 9, because I think 10 has almost bluish color, and 9 is greenish. So I would say 9. So again, this should, if you're familiar with the pH scale, this should tell you right away for your conclusion what the chemical nature of the substance is, whether it's a base or whether it's an acid. So that was glass cleaner. Now, soap solution. This is an interesting one. And the reason why it's interesting, some people might think that soap is, strongly basic substance. Because, all of us, we've heard stories about soap changing the skin's pH, harmful for skin. You don't want to wash your hands too many times because, it dries your skin and whatnot. Let's see, pH-wise, what typical hand soap looks like. So I'm going to add blue litmus, followed by phenolphthalein. By now, we notice that it tends to respond to basic substances quite dramatically. So I'm going to add some phenolphthalein here. And finally, I'm going to add bromothymol blue. let me see. And lastly, we're going to do the universal test, but in a moment. So this is what we're looking at. Now, bromothymol blue, blue litmus, because this is the first one that we're doing, you've seen that it's blue. It's remained blue, but there's a slightly purple hint to it compared to the actual blue of the basic glass cleaner. If you compare phenolphthalein of the soap solution with that of the glass cleaner, you see that there's a marked difference. To me, phenolphthalein produced a very, very faint pink color that you may not even be able to see because of the camera. But it's very, very lightly pink, almost imperceptible. Compared to this, quite a difference, right? That was the glass cleaner. And if we look at our bromothymol blue, you see this is green, right? Does anyone know what color you get if you mix blue and yellow? Like if you worked with paints, you may, know. Yeah, you get green. So this indicator, sometimes with indicators is not 100% this or 100% that. Indicator changes color gradually. So what this tells us that pH-wise, we're somewhere in between the definite acid and the definite base. So we're somewhere in the middle there, and based on the slight pink color of the phenolphthalein, you probably can guess or make an educated guess whether we're closer to the base or the acid end of the pH scale with the soap solution. And finally, yeah, you can use pink for phenolphthalein or light pink, whatever, depending what you used previously for the sodium hydroxide and the glass cleaner. Finally, let's dip our universal paper and compare. So to me, what does it look like to you? 6, 7? 7, exactly. You see that we're almost exactly at 7. Again, this thing isn't perfect either, but it's much better sometimes as some of those other indicators. So soap solution, in fact, has pH very close to 7, and that explains that greenish color of the bromothymol blue, right? Because we're almost at that border point, so it cannot decide whether to change to blue for basic or the yellow for the acid, right? So actually, if you test your soap solution with the pH strip, as we've done right now, your soap, your typical soap, is not that aggressive of a substance. It's been formulated, modern soaps are formulated so that they are very gentle on the skin, and that gentleness comes partly from the fact that it's not a strong base or a strong acid. It's almost neutral. So that's something new, maybe for some of you, at least those who thought that soap is a strongly basic or strongly acidic substance. Let's see what we have remaining. So we have 3 other interesting substances: pop, vinegar, and milk. Again, some of them you may be familiar with more so than the other. 7 is pop, number 8 is vinegar, and number 9 finally is milk in your table. I just picked something that's colorless and clear as much as possible. Next one is vinegar, also colorless liquid with a strong smell. For those of you who, if you pickle vegetables in summer, or if you use it for cleaning, you should be familiar with vinegar. And milk, number 9. Is milk. Yeah, so milk, I've diluted it very slightly so that it's not as cloudy, but it shouldn't change the chemical properties as far as pH testing goes. So let's start with number 7, pop. Same indicators, starting with blue litmus. You already may see the color change to red. Phenolphthalein, no color change. And finally, bromothymol blue, yellow. So I'm not going to lift it up because you probably see the colors already. Red, colorless for phenolphthalein, and yellow for bromothymol blue. Let's dip another one of these pH strips. Okay, so there's definitely color change from what it was before we dipped it into the solution, but let's see what it is. I'm just going to try to rotate. No, you're reading the wrong set of numbers there. Just read the top. Yeah, so I would say this is probably between 3 and 4, maybe, depending how you look at it. 3 probably is the closest match, because 4 already is going into orange, and this definitely has some red to it. So I would say this pH 3, you see that your pop is almost as acidic as your grapefruit juice, right? So also keep that in mind next time you opening another bottle. Your teeth probably don't like the acidicness of pop just as much as that of grapefruit juice. So it's quite relatively acidic substance, as the pH strip tells us. Let's see about number 8, vinegar. Again, here you probably know the answer already, but well, we're going to do it because it's on the list. Again, blue litmus, phenolphthalein, and bromothymol blue. So you see that the color pattern that we're getting with the 3 indicators for vinegar is the same as that for pop. So, it's almost like, if you're drinking pop, it's like you're drinking vinegar or something, pH-wise. Let's see what we get with, see, we get the same kind of response with the pH paper. It's maybe closer to 2, but I think this is still 3. Yeah, I'd say this is still pH 3 because 2 is definitely too red for this. So pH 3, vinegar. So yeah, and we got the same result for pop. Of course, it depends on the concentration sometimes and so on, but it just tells you that chemical nature of pop and vinegar and grapefruit juice, there's something in those 3 that is similar and causes the indicators to respond in the same way. Let me see, milk. So let's add litmus first, followed by phenolphthalein, followed by bromothymol blue. Now, this was unexpected even to me because you see, I'm used to seeing very light pink in milk, but considering that milk is a natural substance, its chemical constitution might change, depending on where it came from and how long it was stored and so on. So what we're seeing is a blue with bromothymol blue, with blue litmus. I keep confusing these two guys. Pink or light pink with phenolphthalein, and blue with now bromothymol blue. So what is it about milk? Well, you may already know the answer, but let's do the pH strip test before we draw any conclusion. I'm just going to rotate it so it soaks up the liquid evenly. Okay, so let's look at the, I'd say this is pH maybe 8. So you see that milk has pH that is slightly different from 7. It's towards the base side of the pH scale, and that is why fresh milk can keep for a short amount of time without refrigeration, without going bad, because it has some of that base property to it that prevents bacteria from growing. Of course, over time, as bacteria grow in fresh milk, they will multiply and they will turn the milk sour, right? So when they say that milk turns sour, it actually literally did that because pH changed to to the other end of the scale, to the reds here, and that causes the milk to go bad. So that essentially concludes our observation part. Hopefully you were able to fill out this table with the corresponding color changes. If you missed some of these, you can ask your classmates, and your job will be to fill out the conclusion column for each of these substances that we looked at. And hopefully you did the prediction before, but if you haven't, you'll need to fill that out as well. And see if your conclusion matches your prediction. Again, there's a third page to this table. So this concludes the lab, or the in-lab part, and I'm going to give the mic